Hello, my name is Taj Allen, and I've been mentoring under Dr. Jim Ban in the Department of Chemistry here at Wichita State University. And the topic that I have been researching is investigating the role of domain 4 of the anthrax protective antigen on stability and immunogenicity. And so the focal point of my research has been about domain 4. And so domain 4 is one of the domains that makes up the anthrax toxin protective antigen structure, which is shown to the right. And so the anthrax toxin PA, or protective antigen, is the major immunogen in the current vaccine that we have out right now, which is called anthrax vaccine absorbed. And so we want to improve the current vaccine and take out the necessity for a cold chain, which basically is the need to have it at cold temperatures at all times so that it can be kept stable and be able to be used at any time. And so all current use of anthrax vaccines show considerable local and general reactions such as soreness, fever, swelling, and other serious adverse reactions. And so, as you can see on the right, so this is the anthrax toxin protective antigen structure. And so we took out domain 4, which I am scrolling over. And so we took it out because we have seen in previous studies that this is the main place for the CMG2 cellular receptor where they bind to each other. So when anthrax gets introduced to your body, that's the main place for it to bind is the cellular receptor CMG2, and then it further attaches to the cell from there. And so we had two main research aims in question. So we wanted to know if domain 4 is deleted, can it still bind to cellular receptor CMG2? And also, if we delete domain 4, will the structural integrity be compromised? So will the stability of the structure change, or will it stay the same compared to the known protective antigen structure versus the mutant protective antigen structure that had domain 4 taken out? And so the hypothesis of the study was basically that without domain 4, that there would be no immune response to protective antigen due to domain 4 having or is being the majority of the immune response to protective antigen. And also the reason why we believe that is because we believe CMG2 is blocking a key epitope or epitopes within domain 4, which is basically just binding domains in domain 4. So if CMG2 is blocking those domains, then it won't be able to bind to the actual cell and make an immune response. And so we used two different primers to cut out the domain 4 out of the protective antigen sequence or genetic code. So those two were PAD for deletion forward and PAD for deletion reverse. So as you see at the bottom, you can see those the genetic sequence for those two primers. And so basically they matched up with the beginning of the domain 4 sequence and the end of the domain 4 sequence so that we knew exactly where to cut it out. And so we had two main methods that we used for this research. So one of them was gel filtration chromatography. So as you can see here, when proteins are put through a gel filtration tube, that you can see that the larger they are, the harder it is for it to get stuck within the small hollow beads that are within the column. And so they come out first in the timeline. And so the smaller they are, the more likely they will be to get caught up inside the small beads. And so they come out later. And so you can use that to separate different proteins from each other. So the larger they are, the faster they come out. The smaller they are, the slower they come out. And the second method was the circular diagram spectroscopy. So basically, all that is we use different wavelengths of light. And so different parts of the secondary structure, which is the alpha helix and beta sheet part of the protein, are absorbed by the different wavelengths of light. So we can see that the different compositions. So if the protein has more alpha helix structures than the previous structure, so if the mutant that we made had a different composition of alpha helix and beta sheets versus the wild type protective antigen. And so as you can see on the graph, you can see the gold is alpha helix, the blue is beta sheets. And so it just shows the different shapes they make on the graph due to the different wavelengths changing. And so one of the first experiments that we did was a gel filtration experiment. So we wanted to see if it would be able to, well, the mutant would be able to reassociate with domain 4 list PA. So basically, if domain 4 by itself, just the domain, would be able to attach back to the protective antigen D4 list PA that we made. And so as you can see here, there's a strong peak for delta D4 PA. There's a strong peak for wild type D4. And then when you put those two together through the gel filtration column, you can see that because delta D4 PA is larger, it came out first, but on the back side of it, there is a peak of association between the two. So we know that they bound or binded together to make a larger structure, which came out together. And so then from there, after we saw that it did bind to D4 again, we wanted to see if it could bind to CMG2, which is a soda receptor. So we believe that it wouldn't be able to, since we already know domain 4 is the main reason why it can bind to CMG2 in the first place. So we put delta D4 PA and CMG2 through the column together, and as you can see here, there's a strong delta D4 peak, but there's no association on the side of it, and CMG2 has its own strong peak, so you can see that they did not bind to each other, and there was no association between the two, which we expected. And so the second part of our experiment was looking at the CD spectrum. So we wanted to see the structural properties and see if they were different between the delta D4 PA mutant versus the wild type PA with all its intact structure or domains. And so as you can see here, they pretty much have the same structure versus wavelength. So we can see that the alpha helix and the beta sheet structures and the secondary structure of the protein are basically indistinguishable. And so we expected that since domain 1 through 3, we didn't mess with the genetic code at all, so it was still going to produce proteins that were still the same as the wild type PA. And so after seeing that it formed a stable structure, we want to look at were the domains still functional. So versus wild type PA versus delta D4 PA, we see that they both unfold at a certain temperature. So in the case of wild type PA, it unfolds at around 51 degrees Celsius, while delta D4 PA unfolds at around 54 degrees Celsius. And so we can see that it's still functional, because if it wasn't functional, then the mutant would just be a straight line across, and that would show us that it was unfolded the whole time and that there's no unfolding, refolding, and functionality to the structure. And also, you can make note of that at around 40 degrees Celsius, there is a unfolding event in one of the domains, and then when it gets to 54 degrees Celsius, the rest unfold, which is not seen in the wild type PA. And so we had to take note of that, since that was different than the wild type PA, and we believe that is due to domain 4 being taken out and not having an influence on the rest of the structure. And so in conclusion, the gel filtration experiment showed that delta D4 has no specificity to CMG2, but it does have association with domain 4, and it can still bind to domain 4, which we expected, since it still had its domains 1 through 3, and the third domain still had all functional sites on it, so it could still reform with domain 4. And then we already knew that CMG2 was not going to be able to bind back with the wild type or the delta D4 PA due to domain 4 not being present, because that is the main site where it binds to, and the epitopes or binding sites on domain 4 are the main points of association between the two proteins. And then the CD spectrum results showed that the delta D4 PA had comparable stability to wild type PA, and so that they matched up as far as their stability and as far as the structure wise. And so then, I said before, the wild type PA had decreased stability at higher temperatures than the delta D4 PA. We believe that is because domain 4 is not there, and we believe domain 4 might have a reducing effect on the stability of the overall structure of protective antigen. And so we believe that without domain 4, that is why we see a higher increased stability at higher temperatures, as seen when wild type PA had a unfolding temperature of around 50 degrees Celsius, while Delta D4 had around like 54 degrees Celsius. That's my literature cited. And then in ending, I would like to thank the McNair Scholars Program, as well as Dr. Band's lab and his mentorship. And I'm now open to any questions. I would say the foremost problem that I faced while doing the research was trying to grow the appropriate amount of protein to do the experiment. So when we do the gel filtration experiments and the CD experiments, we have to have enough protein to put through the column so it can even be picked up by the machines that we use. And so when we grow, it takes around like 2 to 3 to 4 days. And after that, we have to purify it and make sure that nothing else is contaminating the protein so that when we do use it, if we get the results back, we can know for sure that if any discrepancies happen, it's because of the protein's changed structure and not some other protein that's in there that's messing up our results. And so having to kind of time everything and make sure we had everything to begin with was the hardest part. Yes, so the most important part would be the CD experiments that we did, because we expected all the results of the gel filtration, so we knew that we would see some association with domain 4. We didn't know how much, but we knew that we would see something. And as far as CMG2, we knew there would be no association. But as far as the CD spectrum, we didn't know how the structure would look and if it would unfold at all, or if some of the domains would not work correctly due to domain 4 not being there. And so that was a kind of the focal point of our research, to see exactly how the structure is functioning. So I hope to finish up with this research, and hopefully it can be taken to a next stage and a paper being written over it. And so that's the main thing I'm hoping for right now.